Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning sessions. Today we learn about adjuvants in this e-learning platform. The term adjuvant comes from the Latin word adjuvare, which means to help or aid. Adjuvants can be defined as substances that increase the immunogenicity of a vaccine formulation when it is mixed with vaccine or it is added to vaccine. The choice of the adjuvant is of utmost importance as it can stimulate strong humoral and cell-mediated immune response, indispensable for protection against many foreign substances. Non-living vaccines, especially those consisting of small molecules, are not very strong antigens but can be made stronger by injecting them along with some other substances such as aluminum hydroxide, aluminum phosphate, calcium phosphate, albumin, etc. Such substances are called adjuvants. These adjuvants are group of heterogeneous compounds which evoke or increase an immune response to an antigen. The history of adjuvant discovery begins with Gaston Ramon, a veterinarian working at the Pasteur Institute in 1920. Example: mineral salts like alum salts, emulsions, microparticles, saponins, cytokines, microbial components or the products, and the liposomes could be used as adjuvants. So these components could be used or mixed with the antigens or attenuated pathogen to increase the immune reaction. The mechanism of action of adjuvants. The adjuvants decrease the amount of antigen to which the immune response is activated. Adjuvant reduce the number of doses required to induce protective immunity as it increases the immune reaction for very small quantity of the antigen, and this induce protective response more rapidly, and it increases the rate of seroconversion in special populations like exceptional cases like immunocompromised individuals or individuals with chronic diseases, neonates and infants. In these conditions, so it increases the antigenicity of the given antigen. So that is the mechanism of action done by adjuvants. In addition to this, these adjuvants also have some properties, like they have the ability to enable the antigen to be slowly released so as to be recognized by the immune system. So the retention time of the pathogen release or it is recognized by the immune reaction will be, or the immunity will be increased. And this preserves the antigen integrity. Adjuvants preserves the antigen integrity, and this targets the antigen presenting cell and increases the phagocytosis. These adjuvants induces cytotoxic lymphocytes. They produces high affinity immune response when the antigen is mixed with the adjuvants, and they have the capacity for selective immune intervention. So these are the few properties of adjuvants where they increases the immune reaction even at very low concentration of antigens. Coming to the types of adjuvants, there are different types of adjuvants like the mineral salts, saponins, etc. So here in your syllabus, we have included, friends, complete adjuvants and, friends, incomplete adjuvants. So these two are the types of emulsion adjuvants, friends, complete, and another one is, friends, incomplete adjuvants, FIC. Okay. In 1930, scientist Jules Friend developed a powerful adjuvant composed of water in mineral oil emulsion, water in mineral oil emulsion that also contained heat-killed mycobacteria. Heat-killed mycobacterium, that is Mycobacterium tuberculosis. So it is a mixture of water in mineral oil emulsion which has the heat-killed Mycobacterium tuberculosis. So this was invented by Friends. So this adjuvant is called as complete Friends adjuvants. Friends complete adjuvant, or otherwise called as complete Friends adjuvant. It's a water in oil emulsion that contains heat-killed mycobacteria and is a classic gold standard representative of group of adjuvant. It is very strong in making very less concentration of antigen to be recognized by the immune system, and it is a very good adjuvant. Another type is the Friend's incomplete adjuvant is also a water in oil emulsion, but it is not having mycobacteria like our complete Friend's adjuvant. In the 1950s, the use of incomplete Friend's adjuvant as an adjuvant in an human influenza vaccine led to higher long-lived antibody titer. When the human influenza vaccine was developed, the antigen was mixed with the incomplete Friend's adjuvant, and that showed long-lived antibody titer when compared to the same formulation without the adjuvant. That means, so these Friend's incomplete adjuvant increases the high affinity antibody production, and they will results in the long lasting immune reaction. So that is regarding incomplete Friend's adjuvant. So in this session, we learnt about what are adjuvants and what are the mechanism that will be done by the adjuvants, and then we learnt about few properties of the adjuvants and types of adjuvant. So adjuvant is the substance that is mixed with the antigenic preparation to increase the immune reaction. I hope you understood this session. Thank you.